Blast. Good morning to one and all. I am Dr. Tariq, Department of Biotechnology, Islamic College. The bioinformatics is the one of the topic. It's going on the last unit. The tool is Blast. Basic local alignment search tool. This is called a Blast. It is one of the tools using the bioinformatics. In bioinformatics, BLAST is an algorithm and program for comparing primary biological sequence information such as the amino acid sequences and protein or the nucleotide of DNA and or RNA sequences. This is the introduction. BLAST is the meaning of abbreviation of Basic Local Alignment Search Tool. I once again repeat it. Basic Local Alignment Search Tool, that is called BLAST. That BLAST is their main bioinformatics tools, and it is referred to as suite for programs used to generate alignments in between a nucleotide or a protein sequence, referred to as a query, and a nucleotide or protein sequences within a database referred to as the subject sequences. How does BLAST is working? The BLAST works by detecting local alignments between sequences that work the best one. The BLAST computers start with a small set of 3 letters which they call as query word. These letters will be present 3 amino acids or nucleotides in a specific order. For the example, nucleotides ATC in their order. These are all the examples of bioinformatics, that means working tools of BLAST. BLAST அப்படிங்கிறது என்னென்னா ஒன் ஆஃப் த பயோஇன்ஃபர்மேட்டிக்ஸ் டூல்ஸ், அதாவது பேசிக் லோக்கல் அலைன்மெண்ட் சர்ச் டூல்ஸ்ன்னு சொல்லுவாங்க. இது மோஸ்ட் இம்பார்ட்டன்ட் ஃபார் ஆல் கைண்ட்ஸ் ஆஃப் பயோஇன்ஃபர்மேட்டிக்ஸ், பர்டிகுலர்லி பயோடெக்னாலஜி ஆல்சோ இஸ் பிளட் சீக்வன்சஸ்க்கு ஸ்டோர் பண்ணுறதுக்கு, ரெக்கவர் பண்ணுறதுக்கு இது யூஸ் ஆகும். ஒன்று, அது எப்படி ஒர்க் ஆகுது? ஹவு டஸ் எ பிளாஸ்ட் ஒர்க்? ஃபஸ்ட்டு பிளாஸ்ட் என்ன செய்யணும்னா டிடெக்ட் பண்ணும் லோக்கல் அலைன்மெண்ட்டில் எங்கே சீக்வன்சிங் இருக்குது அப்படின்னு பார்க்கும் பெஸ்ட்டு பிளேஸை. அதுக்கப்புறம் கம்ப்யூட்டரில் ஸ்டார்ட் பண்ணதுக்கப்புறம் 3 letters கொடுத்து கொரி வேர்டு அப்படின்னு கொடுத்தோன்னே அது நிறைய மெத்தடில் அமினோ ஆசிட்ஸு, நியூக்ளியோடைட்ஸு, ஸ்பெசிஃபிக் ஆர்டரு, சம் ஆஃப் த எக்ஸாம்பிள்ஸ் ஆஃப் நியூக்ளியோடைடு, ஏடிசி இன் ஆர்டர் டு. ஸோ இந்த மாதிரி இருந்துச்சுனாலும் ஆட்டோமேட்டிக்காக பிளாஸ்ட் டூல்ஸும் இருக்காங்க. த இம்பார்ட்டண்ட் டேர்ம்ஸ் பயோஇன்ஃபர்மேட்டிக்ஸ் எமர்ஜிங் ஃபீல்டு இருக்குது. டேட்டாபேஸுங்கிறதுனா என்ன, டூல்ஸ்னால் என்ன, அதான் இங்கே வரும். த டூல்ஸ் ஆர் தியா ஒன் ஆஃப் த சாஃப்ட்வேர் டெவலப்டு டு பர்ஃபார்ம் வேரியஸ் டாஸ்க் other the stored data such as searches, analysis, submission, annotations. These are all the importance of tools. Actually, the important database and the important tools, there are 2 different types of work. One of the tools, one of the databases. The databases like NCBI, EMB, LDDB, GenBank, Swisspot, PDB, PubMed, etc. etc. The tools of the Bioinformatics BLAST. This is the first one, basic local alignment search tool. Actually, the BLAST search and homology tools. FASTA is one of the search and homology tools. The summation of tools, the ORF analysis tools, retrieval tools, database taxonomy, summation of tools and DDBJ, multiple sequence alignment, protein secondary structure comparison tools. These are all the importance of tools in bioinformatics. The BLAST is stand for Basic Local Alignment Search Tool. The BLAST is a program which uses specific scoring materials matrices for performance sequence alignment similarity and variety of sequence databases to give us high scoring upgraded sequencing and segment amount related sequences. The compound, the BLAST algorithm and the retrieval faster. Actually, the picture shows that the pictorial method. The number of DNA and protein sequences in public databases is very large number. NCBI protein databases has 38 crore 50 lakhs protein sequences. Searching a database involves aligning the tools of query sequence to each sequence in the databases. For example, predicted protein and alignment A and C. These are the database sequences. And finally, the databases. Types of BLAST algorithms. These are the types of the algorithms. And another one is the explanation of the each and every one. The special types of BLAST. Make specific primer, search, findings, finding search, search sequences, etc. These are all the special types of sequences. Next one, how BLAST work, how it is work. Although how the title complicated with lengthy, so in short and brief explanation, BLAST works in following two steps. Number one is BLAST first researches for short regions for a given length called as a word and subscribe that score at least t when compared to the query sequence that align with the sequences in the database target sequences. And second point, for every pair of sequences that have a word to word common, BLAST extend the alignment in both similar than a certain score threshold. These alignments are called high scoring pairs of HSPS. The maximal scoring HSPS are called MSPS. This is the how to BLAST works pictorial representation. This is the picture method. The word subsequences of the query sec. So these are all the structure of the BLAST method. Finally, the slide shows that BLAST website, the parameters and the values, databases, and optimization, maximum target sequence, short queries, so etc. Any doubt you ask to me through the via mail, WhatsApp. Then example to understand the working of BLAST. The answer is the question arising. We use BLAST for searching the database and then analysis the result which is produced. Here the example, this will seen an example. We have following sequences of a protein from our experiments with a mycobacterium, tuberculosis, etc. The sequence is above one. These are all the BLAST using the sequences. Finally, the summary of the BLAST topics. The BLAST is the most important program in bioinformatics, maybe of biology. The BLAST is based on sound statistical programs, a basic principles and a understanding of its principles is key for interpreting BLAST output. BLAST can play an essential role for helping us to purpose the structure of protein, functions of protein sequence, relation with an organism. Use BLAST or megablast for DNA. Use PSI BLAST for protein searches tools. So these are all the major source of the best searching tools. BLAST, basic local alignment search tool. This is a common sequencing database structure. It can be used, and you have to develop and any kinds of microorganism, and you send the sequencing method. It will be developed and entire printout will be displayed. These are the one of the basic method. Okay, thank you one and all.